DPD Deficiency, supporting the introduction of national pharmacogenomics services for testing. And this is hosted by Whitehead Scientific and Yourgene Health. Before we start, I'd just like to mention some housekeeping. There is a chat box available where you are welcome to post your questions and comments. And we will have a question and answer session at the end where we will address these. The recording will also be available and will be shared to all attendees. So the agenda of the webinar is as follows. I myself will be giving a brief overview of Whitehead Scientific, and then I will hand over to Louisa Ive at Yourgene Health to tell us more about DPD deficiency and testing. At the end, as mentioned, there will be a time to answer your questions. So today, I myself will be introducing Whitehead Scientific. My name is Jacques de Pre, and I am the Clinical and Automations Product Manager at Whitehead Scientific. Louisa Ive will be doing most of the talking today, and she is the Clinical Product Manager at Yourgene Health. So first of all, an introduction to Whitehead Scientific. Who are we? Whitehead Scientific is a leading privately owned distribution company that supplies instruments, reagents, and consumables to the South African market, or Southern African market for that matter. Our motto speaks for itself and reads products, expertise, and support. We were founded in 1988 and currently have a team of 37 people. We cater for products in the fields of clinical and automation, molecular and cellular biology, as well as lab essentials. We have a large portfolio of carefully selected suppliers offering only the highest quality of products, ranging from plasticware up to large benchtop instrumentation and cold storage solutions. So recently, we are very proud to say that we have added Yourgene Health to our portfolio of reputable suppliers. And without further ado, I will hand over to Louisa. So Louisa, I'll stop sharing my screen and then you can take over.
Perfect, thank you. Let me share this here. Fantastic. So do let me know, are you able to see that, just to check? Don't know if anyone from Whitehead can confirm.
I can see, yes. I can see the screen.
Perfect, amazing. Okay, so perfect. Well, thank you for the introduction. Really fantastic to be here today. As was kind of said in the introduction, the plan for today is to present to you both our DPYD genotyping product that we have at Yourgene Health, but also to discuss some of the wider conversation that's currently ongoing about the clinical utility and implementation of this testing in routine oncology workflows. And I really also wanted to use this time to highlight some of the existing partnerships we have ongoing to deliver this kind of routine testing at a national level in a number of countries. So I won't spend too much time talking through who I am or who Yourgene is, but just to give a bit of a background, as Jack said in the introduction, I'm the Clinical Product Manager here at Yourgene Health, covering the PCR range of products, which includes our DPYD assay. I'm also actually a trained clinical scientist, and I previously worked in the NHS here in the UK for a number of years. And Yourgene Health, we are a leading molecular diagnostics manufacturer covering a range of products and diseases, including reproductive health. So as I said, won't spend too long talking about who we are as a company, but Yourgene Health are a leading molecular diagnostics manufacturer. We have our non-invasive prenatal testing suite of products, but we also have products that cover cystic fibrosis, male factor infertility, and of course DPYD, which is what we're looking to cover today. We have a really busy and active R&D department that's keeping us at the forefront of innovation, as well as a service lab offering as well. We globally have a presence in over 60 countries, including the US, which sees us active in many and quality and regulatory spaces, including obviously CE-IVD. Our head offices are actually in Manchester, but as I said, we have a presence in Singapore, Taiwan, Canada, and the US. So that's just a little bit about our US partnership there as well. So to begin, what is DPYD testing and why are we concerned with it? So what is the story of clinical utility here? So the DPYD gene codes for the DPD enzyme, which is in itself important in breaking down and metabolizing drugs from the family classed as 5-FU agents. So 5-FU based chemotherapy is used in over 2 million patients per year globally. And as you can see, you know, that is covers a huge range of common tumor types. So the point is that this isn't really it's not a niche drug. It's really commonly used in over 2 million patients per year, and you know, this is big business, and so there's a huge coverage here and a huge potential to have a lot of impact if we work in this space. So we know that fluorosil and capecitabine are fluoropyrimidine products widely used in the chemotherapy treatment of patients with, as I said, a wide range of tumors. So you've got gastrointestinal tumors, head and neck, breast, and other forms of solid tumor cancer. And as I indicated, the DPD enzyme is necessary to catabolize those chemotherapy agents to inactive products. And what we're doing when we catabolize that is we're limiting the circulating levels of the drugs. So what you have when DPD enzyme activity is deficient is patients that are treated with a standard dose of those drugs, but they're at a significantly increased risk of severe toxicities. And by severe, I mean grade 3 or higher. And I'll talk more about that in a few slides' time. But actually, there is a significant risk of death here as well. But what we know is that DPD enzyme deficiency is strongly correlated with certain variants in the DPYD gene. And that's actually those listed in the CPIC guidelines. And again, I will come back to that in more detail later, but you can see that the Clinical and Pharmacogenomics Implementation Consortium there, they're very instrumental in this kind of setting the guidelines here and recommending dosage modifications when somebody has a deleterious genotype. I think some studies have also found that toxicity in these patients sits at around 40%. But what I think it is really important to state as well is that not all DPD deficiency and not all associated toxicity is associated with that deleterious genotype. So that's to say there is a portion of patients who experience toxicity and are not found to have a mutation and therefore wouldn't be identified in the preemptive approach we're discussing today. But we can talk more about that later on. And also that lends itself to thinking about the positive predictive value here as well. But what I wanted to emphasize on this slide was the actionability. So in those individuals with a variant identified by one of those variants listed in the CPIC guidelines, so evidence level A, as you can see on the slide there are recommended dosage modifications. Or actually in the case of complete deficiency, for example, the MHRA, which is a regulatory body here in the UK, advised that patients shouldn't receive this type of chemotherapy. So actually what we're seeing there is not only a recommendation, but actually a contraindication. I think it's important to note, and I will talk more about kind of international testing guidelines and recommendations later on, but the FDA drug labeling doesn't quite go as far yet as to provide a warning about the safety in complete deficiency, but they do have a table of associations published last year that does make recommendations based on dosage. And again, I'll cover that in more detail later on. But really, the concluding point of this slide is that there is, when you have an individual who's a carrier of a mutation treated with a standard dose, there's this significant risk of toxicity, but there is a pathway suggested here. So this is an actionable test. It's not a test just done for information purposes. There's an output at the end of it. So I guess if we turn our attention a little bit more to that clinical utility, as I said, so let's dig a bit deeper into that. Why are we doing this? Why are we interested in this testing? Well, when we kicked off this presentation, I kind of indicated or alluded to the toxicity associated with 5-FU and these patients with one of these deleterious genotypes. But how does that actually present? Well, what I'm showing on the slide here is that this is a really multi-system severe reaction involving the skin. You've also got hematological deficiencies and gastrointestinal side effects, as well as problems with the heart and nervous system. So actually, in someone with a DPYD mutation, the risk of experiencing that toxicity is as high as 88% in some studies, and that in itself confers a 10% risk of death. And if we just circle back to, okay, so that's for those at an individual level, there's a very high risk if you have one of these mutations. But how many people across the globe are having these mutations? What sort of burden is this mutation or this mutational range having? Well, we know estimates sit between 3 and 8% globally, depending on obviously which population you're looking at. And DPD deficiency, so that's encompassing all of those individuals with a deleterious genotype, but also those who maybe don't have one of these mutations, but do have an enzyme deficiency. If you consider all of that group together, the figure then sits at about 12%. So this is not, yeah, as I said, this is not a particularly rare condition. This is common, and that lends itself obviously to thinking about some of those studies of clinical utility and why you can have such an impact with doing this testing. So not only at a personal level, but at a population level as well. And just to touch on those CPIC guidelines I mentioned before, the Clinical and Pharmacogenomics Implementation Committee. So as I said, what they've done is they've looked at some prospective data and they've looked at the DPYD genetics, and they've come up with a series of mutations that they feel there should be dosage adjustments for. So you can see here on the slide, there's the 3 allelic variants, but then you've also got a haplotype, which you can test for by way of any of the associated mutations, if you like. But yeah, so 4 genotypes, if you will, but actually 6 variants there. And what you can see at the bottom of the slide here is that what the CPIC guidelines have done is they've taken those genotypes and they've converted them into so-called activity scores. And those activity scores will give a likely phenotype. And based on those, you can think about doing dose reductions. So just to emphasize the point again, that in individuals who are poor metabolizers, so that would be an activity score of 0 or 0.5, they should not be given 5-FU chemotherapy. So that's a contraindication there, and an alternative drug should be considered. I think, the CPIC guidelines are really the sort of central point of discussion here, and kind of how we've designed our assay, that I'll come to talk about the product itself later on. But what other guidelines are we looking at internationally? Well, I don't need to read off the slide here for you, and this is by no means a comprehensive list of recommendations. But I think, what you can see here is the NCCN, the National Comprehensive Cancer Network, which is obviously a huge network and very influential in terms of policy. And they have some guidelines for colon cancer, stating that certain carriers of DPYD variants have a significantly elevated risk. So in their guidance, they do make reference to this. When we kicked off the presentation, I mentioned in some of the intro slides about the FDA. So just to say, the FDA does acknowledge, as I said, that DPD deficiency is a risk factor for toxicity, and DPYD is listed as one of those valid biomarkers. And last year, in February, the FDA published this table of pharmacogenomic associations, which lists sort of drugs and genes and those interactions which may be of relevance. And 5-FU and DPYD were listed in this table of associations as having a higher adverse reaction risk. But they haven't yet gone as far as to recommend testing or require it prior to drug initiation. But I think it's fair to say that this is going to be something on their radar over the next few years. So I wouldn't be surprised if this changed, I feel. So the European Medicines Agency, actually, this is interesting. The European Medicines Agency does recommend testing of all patients prior to treatment. And actually what's interesting about this is that it was implemented after a campaign group from a patient organization in part. And so, yeah, that was really driven by the patients themselves and then widely adopted. Interestingly enough, in Canada, just to call out there as well, in the province of Quebec, you have a widespread adopted practice of DPYD genotyping prior to treatment. I think just to call out a little bit about the NCCN again, in 2018, as of that point, they continue to state that universal pre-treatment testing of all individuals remains so-called investigational for insurance reimbursement purposes. But I think, this just highlights again, there's this kind of discrepancy between what is beneficial at an individual level. I don't think any of these organizations are arguing that at an individual level there's any argument that clinical utility is not established, but it's just about whether at a population level this makes sense. So I think, there's obviously always going to be a slight tension there. So I wanted to just touch on the evidence of this clinical utility. I hope just from reading this slide that it's really clear that there are some really truly large scale studies that have found time and time again that testing is clinically useful for these patient groups. So the impact of pre-treatment genotyping is absolutely huge in terms of toxicity reduction. So you can see here in this study from 2016 that genotype guided dosing reduced severe toxicities from 73% to 23%. So obviously a huge impact for those individuals who were saved from that toxic reaction, but also, you know, a saving for the healthcare providers themselves as well. There are, you know, aside from this, from what I've listed on the slide here, many other sample studies and sort of examples where this has been proven. So I think actually even as early as 2008, studies were showing that patients who received 5-FU dose adjustment based on pharmacokinetic monitoring showed significantly improved objective response rate, as well as a trend to higher survival rate and fewer of those grade 3 and 4 toxicities, which is what I was talking about when I showed that slide about the sort of multi-system reaction that you're looking at there. I think I will highlight on a later slide. I'll touch on economic utility, because I don't think you can ignore the other side of the coin, if you like, here. Just because something's clinically useful, obviously you have to think about resources and you know, in an ideal world we wouldn't have to do that, but I think it's important to touch on. But as I said, we'll come to that later. But what I wanted to just kind of highlight as well in this study is that matched analysis from actually the same 2016 paper, but also a 2019 paper. They found that there was no reduction in response or survival when dosing was lowered. And I think that's a really, really important point to make, because although clinical utility could be established in terms of the studies that have shown reduced toxicities, what many people were questioning is, okay, well, you have fewer toxicities or less severe reactions, but what are you doing in terms of the response to the chemotherapy itself? Are these individuals receiving a lower dose? Are they getting the benefit of the chemotherapy in terms of their tumor response? And yeah, what these matched analyses have shows is that response and survival is not impacted. And I think it's important to call out, because clearly what we're looking at here is a reduction in dose, but consistency in systemic levels of the drug. So you're not actually reducing the impact of the drug itself, you're just preventing that overload, and enabling that metabolization to be safe. I didn't think that I could to go through this presentation without touching on the fact that DPYD genotyping is absolutely not the only way you can assess for DPD deficiency. I think I mentioned in the first slide, what we see is sort of a portion of patients who would have a normal DPYD genotype, but actually they would have one of these toxicities. And one argument for that is that, you know, there's a discrepancy. Not all DPD deficiency can be explained by an adverse genotype. So I just wanted to touch on that on this slide here. So several studies have actually aimed to ascertain DPD activity itself, like phenotypically via observable sort of variables. Examples of this would include enzymatic testing of hepatic biopsies and measurement of peripheral blood mononuclear cell activity, so PBMC. Some of you may have come across that in the literature or in clinical applications. However, there has been shown to be a poor correlation between PBMC DPD activity, hepatic DPD activity, and systemic clearance. So this really casts doubt on the clinical relevance of this method. And I also think it's important to point out that using this testing approach is costly and invasive for patients. I think, you know, also a piece of evidence we can't ignore here is that there is a lack of CLIA, that's a US regulatory body for laboratories. There is a lack of CLIA approved availability for enzyme testing, which has led to slow uptake in clinical practice in the US. So if you think about the genetic testing registry, the only CLIA approved tests for assessing DPD deficiency are those genetic assays themselves. You know, either analyzing the entire coding region, deletions, duplications, or as we're talking about with our product here, those targeted variants of DPYD, most probably aligned to those CPIC guidelines. So, you know, you can think about DPD activity. The jury's still out there as to whether that's a standardizable and safe approach for patients. However, the benefit of TDM over, so if you think about therapeutic drug monitoring as well, which is kind of the next arm, if you like, working down from DPD activity towards genotyping. So that's therapeutic drug monitoring itself. Therapeutic drug monitoring, obviously, whereby you measure actual systemic clearance of the drug rather than a proxy measurement. The benefit of that over either DPD activity or DPYD genotyping, which is what our product lends itself to, as I've said, the benefit of the therapeutic drug monitoring is that you directly connect drug levels to clinical outcomes, with continued follow-up allowing for feedback and that sort of a dose adjustment loop. However, TDM actually carries the risk of first cycle toxicity. So what I mean by that is you don't fully eliminate the need for pre-treatment screening for DPD deficiency. So you can't monitor, I mean, it sounds self-explanatory, but just to make the point, you can't monitor for a drug until you've given the drug. So in those individuals with a DPYD adverse genotype or DPD deficiency, they would still have this toxic reaction after one cycle. So you aren't fully eliminating the risk for those individuals. And as we saw, that risk is not insignificant at 88% for a severe reaction and 10% risk of death in some studies. Other drawbacks of TDM are difficulties in standardizing this approach, and actually the inherent costs of employing such an intensive program. There's actually a really nice review from 2016, which I haven't put the reference on this slide, but I will make sure that, if anybody's interested in that, I can follow up and send that over, which is, a really nice review that covers DPD activity, TDM and genotyping, and compares and contrasts all of the different approaches and the relative benefits and drawbacks to each. But actually, I think just a concluding point, as I'm showing on the slide here, is that in prospective trials of DPYD genotype guided dosing, there is support for the widespread implementation of the genotyping approach over and above activity monitoring or therapeutic drug monitoring. I think just to touch, I know something that comes up when I've talked about this subject with people before is people ask about this My5FU, the Saladex pharmaceutical product, which is a biochemical assay using nanoparticle technology to determine those circulating levels of 5FU, so a form of therapeutic drug monitoring. I think arguably there obviously are some advantages for this approach. So you can see on the slide here, there is a 15-minute turnaround time for this approach. But just really, again, to emphasize, not only is there that risk of first cycle toxicity, but there is also a cost implication here. So NICE guidelines, which is obviously the body here in the UK that conducts these sort of healthcare economic analysis for any intervention or anything that's being considered, estimated that the total lab cost per blood sample for using My5FU as a TDM approach was about £25. And this is, this immediately stands out to me as, a very reasonable figure. But actually, the point I wanted to make is that you need to do this blood measurement time and time again, because as I said, you need that feedback loop to be doing your dosage adjustment and making changes. So actually, repeated measurements would be needed. Whereas if you think about genotyping, which is not a million miles away in terms of a pricing benchmark from that £25 estimate, this genotyping is clearly a one-time test. Your genotype doesn't change, so you do the test and you have the result, and you can make your dosage adjustment and go from there. But perhaps there is a role for all of these tests to work in complementary manners in some healthcare systems. It just depends which benefits and drawbacks are of utmost importance for depending on the provider. But I think, as I said, the literature is typically in agreement that those DPD phenotyping tests in general require further validation of clinical validity and utility. It's actually interesting, and one authority that I wanted to call out was actually the French medical authorities, and they began requiring certification a while ago, actually, that patients are free of DPD deficiency before giving a 5-FU based chemotherapy. And I think this has been effective since 2019, actually. And the standard test that they recommend, or the standard test that's in use in France, is plasma levels of DPD activity as assessed by uracil measurements and dihydrouracil as well, I think. And the application of this in a medical setting has been reported. But yeah, I think, I think it's the jury is still out as to whether the validity and utility is established for those approaches, whereas I think genotyping is universally understood to, of course, have drawbacks, but the benefit is very clear, and the standardization, crucially, is very obvious and very easy to implement. And so to that end, I just wanted to spend some time talking about our assay itself, how it works, just to give you an idea of those benefits, as I said, and how easy it is to implement and standardize your approach for these patients. So what we have is an assay that works based on arms-based PCR. So I don't think, based on this audience, that I need to go into too much detail about the underlying technology here, but this is a very well known and understood, but ultimately very simple approach. So arms-based PCR, we have 2 tubes. We have in the mix A, the mutation primers, except for actually what you'll see here is that there is one wild type primer in the mix A tube, just for control purposes. But again, I can talk about that later on if it's of interest. And mix B covers the wild type primers. So as I said in an earlier slide, we designed our assay to cover the CPIC mutations and CPIC mutations alone. And what we really felt here is that the reason for doing that and for not including perhaps some other mutations is that we wanted to ensure that the output of this was, as I said, actionable by labs. You get the result, you have recommendations on the dosing changes that you need to make or that you're suggested to make, and you know what you're doing with the answer. So there's no uncertainty there around, perhaps whole gene approaches would give you variants of uncertain significance, which, arguably perhaps would increase the sensitivity of your testing, but you're left with some uncertainty about what to do with the result. And also, this approach is very quick, it's very easy, and you're looking at a specific set of primes. Sorry about that. But yes, as I said, the selected DPYD markers conform to the CPIC guidelines, and the sample type is DNA extracted from whole blood. So why would someone choose our assay? Well, the 2-tube format makes it really simple to use, but mostly what I wanted to highlight is how our assay fits perfectly into existing workflows and capabilities. So you don't need any extra equipment or hidden costs. You probably already have absolutely everything you need for this assay. And we're compatible with the commonly used thermal cyclers and the capillary electrophoresis setup. Currently, we support interpretation via genemapper or gene marker. Again, I can talk more about that on a later slide. So we have a 4-hour workflow with very minimal hands-on time, under to 30 minutes typically. So this makes it a very good choice for an oncology setting, where obviously quick results are of absolutely utmost importance. There is a patient waiting at the end of the result to have their treatment. So what we wanted to ensure is that we can fit into typically the labs we work with. Currently, they're looking at sort of a 1-day turnaround time, perhaps up to 96 hours. But yeah, as I said, the workflow itself about 4 hours and 30 minutes hands-on time. So yeah, super easy to integrate. I think DNA extraction wise, we're not prescriptive here. We basically, you can use whatever you like as long as the purified DNA is of the specified quality. This test is super forgiving because it's obviously very simple to use. And I think that's the point just to highlight is, again, just what we're trying to do here is we're trying to create a product that fits into your existing workflows, and you can get up and running super quick without, very onerous requirements to procure extra equipment or extra reagents, or actually, what we've heard from existing users is the training requirement is very low. People are familiar with the systems, familiar with the technology, and able to get up and running really, really quickly. In terms of the validation wise for the PCR platforms that we're looking at, We did this on the ABI series as shown there, but you can use a wide range of thermal cyclers. So yeah, again, as with the DNA extraction, we're not prescriptive here. Probably you have one of these instruments existing in your lab already, so you'll be ready to use and familiar. And we have a fantastic technical support team who can help you get up and running if you do have any questions about using any alternative PCR platforms, or if you did encounter any difficulties in getting set up and going. In terms of the capillary electrophoresis, the fragment analysis platforms that we have, so as I said, we are compatible again, as with the PCR systems, with a range of applied genetic analyzers. The ABI series there as well. Really excited to share actually that last year we validated the kit on the Seq Studio, and that has been fantastic for many of our customers. So that's enabled them to use that instrument, which they were clearly very keen to get up and running with, and use transfer over their existing assays. As I said as well, we are compatible with both genemapper and genemapper. We have all of the analysis settings available that you could possibly need on the website when you get going. And as I said, our fantastic tech support team are always on hand if you do encounter any difficulties, or if you need help getting set up. So hopefully the workflow is very clear there, standard arms-based PCR technology, but I wanted to just spend a few slides showing you some example results so you can have an idea of what the output looks like, and really just to highlight how simple this is to interpret. So what you see here on this slide is, is on the as I said, the mix A is obviously the mutant primers, the mix B is the wild type primers. But what we do is we include one of the wild type primers in the mix A, as I said, because what that does is it acts as a control for you, so you can be sure that amplification has occurred. So yeah, for this particular mutation, the wild type and the mutant are in both tubes. What we're looking at here, clearly all green peaks, A and B is a wild type result. What you can also see is the red peaks here between the A and the B mix, just indicating those STL markers. So for your patient identification, just ensuring that you've got the same patient in the A mix and the B mix. Obviously, you wouldn't want to be mixing up patients in that way. So that helps as another quality control step. This particular patient here is a heterozygous result when you've got, you can see one blue peak here indicating a heterozygous result for this particular mutation. And then the wild type results in green again for the other mutations, with this for this particular mutation being in mix A, but the rest of them being in mix B. And finally, just to give you an idea in terms of the peak height of the difference between a homozygous result and a heterozygous result. Obviously, what you're seeing here is that for the 2846, for this homozygous individual, there is not a corresponding peak in the B tube because they don't have any wild type material to amplify. So hopefully, hopefully that's very simple and gives you an idea of exactly how easy it is to analyze and interpret these results. But I think it was important just to touch as well on the performance characteristics of the assay. I appreciate this is actually quite a busy and noisy slide, but I really wanted to show that we've done robust testing of this assay, and we wanted to make sure that it obviously works compared to the gold standard of Sanger sequencing, or in terms of the overall percentage agreement. And yeah, what we're looking at here is 100% overall agreement in terms of the positive and the negative results. So in terms of proxies for sensitivity and specificity. So when we've Sanger sequenced a sample and we know what we're expecting and we've compared with our kit, 100% concordance across the board there. What we are also looking at is precision and reliability. Again, 100% across the board for a range of different mutations and sample types. As I said, if anybody's interested in scrutinizing this data in more detail, always happy if you get in touch after the webinar, I can send these over. And they're all actually listed in the IFU as well. So if you were up and running, you'd be able to browse these at your leisure. And precision and reproducibility as well. Again, 100% across the board. So I said when we were talking about clinical utility that I thought, it was necessary that we covered economic utility as the other side of the coin. We can't ignore this. It can sometimes feel uncomfortable to talk about the price of testing and the relative value for a healthcare system, but unfortunately for the vast majority of healthcare systems, and resources are limited, and there always will be, and there always perhaps arguably should be, a question of, is any test or intervention economically viable at a population level? So just to give you an idea on the slide here, there are, again, as with clinical utility, a number of large-scale studies that are looking at different study designs and populations and showing across the board economic utility. So you can see there's 3 papers here, one from 2016 and some from the last few years, showing that upfront genotyping of all patients has been demonstrated to be either cost neutral or cost saving. I think that's an important point. It's not always about saving money. Sometimes if something is cost neutral, i.e. it doesn't cost you anything, but it has a clear clinical utility, then again, that's a no-brainer in terms of implementation as well. You can see here, get an idea of the average cost of an ICU admission. And actually, I wanted to stress as well that this figure has been expressed in dollars. This was actually in a European healthcare system, I believe. So I think if this was dollars in the US, this figure would be even higher. NCCN, again, they've done wide-scale prospective studies. I think that's important. This is not just retrospective, but they've looked prospectively and found it to be cost-effective. And in NHS Scotland, where we've got one of our partnerships that I'll talk about in a few slides' time, this is screening is less costly than a no testing strategy. And actually, I think the other thing that I haven't listed on the slide here is I just wanted to indicate that there is actually there is an antidote for 5FU toxicity licensed in the US, a product or a medication called Vistagard. We could spend a long time talking about Vistagard. I won't go into too much detail about it here, but Vistagard, it has to be administered, and there's a very short window of time for it to be effective. But moreover, a single dose of Vistagard costs $80,000. And if you consider that compared to a simple genotyping test, I think the economics of it speaks for itself. And finally, just a slide just to talk a little bit about our partnerships that we have here at Your Gene Health. So what we've seen over the last year or so is institutes beginning to use our assay to deliver a pilot program. And then following this is a real ramp up in terms of national and governing bodies putting this evidence into practice with solid recommendations, and importantly also funding for all patients to be tested before having this type of chemotherapy. So currently, as we speak today, the current status now is our assay is being used to deliver this testing, as I said, across a number of sites internationally. But just to call out a few here in the UK, so we are the sole provider of testing for Wales. We have a huge presence in England across NHS institutions there. In Belgium, just to call that out, our assay is actually being used in the key institute, the institute that was actually leading on the national level recommendations, which they published either earlier this year or late last year. I can't remember exactly when that was, but fairly recently they came out with that paper, and we were in use to generate some of that data. So that was a really great moment for us. And we actually really feel this is just the beginning as well. Something I also wanted to highlight as well is that we have a partnership with the 5FU Alliance, who are a patient organisation group here in the UK. They are looking to widen access to testing and get really people talking about this subject. And we're actually really committed not only to making the product available for use, but ensuring we support patient and healthcare providers to keep the pressure on where governments or funding bodies have not yet committed to offering a service. So you can see actually if you go to our website, a recent white paper that we published in conjunction with some leading clinicians and scientists in the field, and the 5FU Alliance were involved with that as well. And it gives a really fantastic holistic view of this issue from different perspectives. And I think the wide-scale adoption of our technology is actually really testament to what we hoped to achieve in the design of it. We wanted something, as I've said throughout this presentation, that delivered actionable results, but importantly was also easy to roll out and scale. It's really crucial that this test fits into existing care pathways, particularly in regard to turnaround time. And clearly a rapid result is important as I said, there's a patient waiting for treatment. The success of these partnerships is really driving the direction of our business as a whole, actually, as we move further into pharmacogenomic testing and precision medicine. And we really do hope to see continued success to the benefit of many, many more patients. And what I've just put on the slide here is finally on the right, a quote from a scientist at one of the labs in England, just highlighting again how easy they've found to implement this test and put it into routine practice to the benefit of patients. Ultimately, that's what we're all looking to achieve. So that was it from me in terms of the core presentation, but absolutely happy to take some questions either now or, I'll make my contact details available so I can always follow up as well.
Thank you very much, Louisa, for that very informative presentation. And thanks once again, everybody, for joining and for your attention. As Louisa said, we'd like to open the floor for any questions that you might have. Please type those into the chat because everybody will be muted still. If you don't have any questions or give some time for questions, you're always welcome to reach out to us. And we will also, as mentioned, share the recording, with everybody that attended.